Well, it's great to be invited to talk at this virtual symposia. It would have been nicer to be there. Been trying to get out for last couple of years, and with COVID, it's going to be a little bit more. But it's great to be able to share the work that the lab has been doing for the last 5 or 6 years to try to really explore the dynamics and genetic regulation of the skeleton in fishes as models for tetrapods, and to what extent we can do that, and to what extent it falls short a little bit. And so this talk, I think, nicely integrates with some of the talks we heard this morning about scoliosis, as well as several of the talks yesterday about osteopenia and brittle bone. And so the major driving force of the lab is to try to understand morphological complexity and developmental mechanisms that may actually potentiate the generation of form and novel structures. And we focus on fishes because primarily, well, let's say teleost fishes, because we're all bony fishes. We focus on teleost fish because they're so diverse. And here is just some X-ray scans of a diverse spectrum of teleost fishes, showing the wonderful diversity in cranial as well as body primary axis patterning that you can see within teleost fish. So the question we would have is, what are sort of the genetic instructions that allow for such variation? And can we harness this in terms of trying to understand both the etiology of diseases in humans, as well as potential therapeutics that actually may not be seen in humans, but we actually have the latent programs in which to activate because of evolutionary relationship amongst all bony fishes. And we focus primarily on the zebrafish as a model, and that is because we can do high throughput genetics and forward genetic screens. So the tale I'm going to tell you today involves two different systems. One is for genetics and one is for genomics, and I'll tell you a little bit about both. So the first part of the story is about genetic screens we've been doing, focusing on post-embryonic development. Now, most screens in zebrafish focus on the early larval stages, we can see here on the left. And this is wonderful because we can go through thousands and thousands of embryos and look for morphological differences. And most of the organ structures of vertebrates start to form at about 24 hours. And so you can quickly go through and actually find unique alleles resulting in developmental abnormalities. And this has been a very powerful approach. But the skeleton actually doesn't form at this stage. The skeleton forms a little bit later. And so the vast majority of alleles that affect the skeleton were not coming out of these screens. And so about 15 years ago in Tübingen, Germany, we started a screen looking at post-embryonic development. So that means looking for adult phenotypes. So these were things that you expect to see swimming around in the tanks. All the earlier mutations are going to die off. All the mutations found in the early German and Boston screens were pretty much lethal. And so these are things you will not see in the screen. Rather, you're getting a select type of mutations that come out that result in viable change in morphology, and oftentimes they're fertile, so that helps the screen as well. And so we do this, and we both looked for recessive. That was about a 4-year screen, and when we came to Boston Children's Hospital and Harvard Medical School, we did a dominant screen, and that looks at the F1 generation as put on the left here. And what we found is that you can pull out a large number of qualitatively distinct phenotypes. And so these weren't like a little bit of growth here, a little bit of growth there. These were monsters, and they were viable. And when we started to look at these, what we've learned over the last 10 years, as we started to clone out the mutations and see what genes are in, is a lot of these genes are things that cause human skeletal dysplasias. And so there's a parallel of the type of selection for alleles and things that affect skeletogenesis that you're going to see both in adult fishes as well as that affects human patients in the clinic. So the question is, can this then be model for testing, for example, small molecules to affect skeletal growth and repair? And that has been basically the focus of many labs. For example, you hear a talk today about osteogenesis imperfecta and trying to figure out regulation of that using this model. But what I want to tell you today is a story about the primary axis, because this actually was an area that sort of felt like it needed some resolution and analysis in terms of the mechanism involved patterning. Well, this is what basically one of these screens look like. You get all sorts of interesting phenotypes coming out. You get short bodies, you get messed up heads, they look like battering rams. You get changes in fin, absence of fins. For example, I don't know, I can't see my pointer here, maybe you can. Short, basically a shrimp coffin, short head fish. So we needed to start to parse this out in different groups to understand about how different mechanisms are working. And so the way you do this in a forward genetic screen is you set up classes of phenotypes, and then ask if all the genes that come out are involved in a similar pathway. And so the first story I'll tell you today about the forward genetics is about patterning of the spine. We create a class of dominant mutants with vertebral defects, and these can be seen just outright because they have a shortened spine. They're called stumpy fishes. And don't worry about all the insignias here, the DMH. This is just the allele. This is how we actually track which genetic mutant we have. And all these are heterozygous, and all of them have a viable change in morphology. They're just smaller. Well, this leads to very distinct changes in vertebral patterning. And the fact that when we clone all these out, what we see is that you get changes in basically the differentiation and formation of osteophytes, and a stenosis of the vertebral column here in micro-CT of an adult zebrafish. We also get cases of patterning that I'll talk about today. We also get cases of compaction. So they arrived at this shortened phenotype by different mechanisms. But when we clone it out, every one of these mutants pretty much was a dominant mutation, a single missense mutation in a glycine residue in a collagen protein. And that can be seen down on the bottom right. Basically what this means is that through different alterations extracellular matrix, we can get different types of emergence of dysplasias that have certain characteristics to them, but they all lead to basically a dysfunctional patterning of skeletogenesis of the bone. One of these cases, CMN down here, is a case I'll talk about next. But we continue that and we get further phenotypes. For example, you get sort of weird compaction and weird sort of hemicentric form, and these are also in collagen as well. So every mutant that we pulled out so far seemed to be fall within collagen biosynthesis and function of making fibrils. And so all these are basically really wonderful models for human disease, osteogenesis, and perfecta. And you've heard a little bit about this earlier in the talk with this zebrafish model that came from Chen Fisher's lab. But what we see here is we have a diverse spectrum of different phenotypes coming from distinct mutations in glycine residues. And all of these can be used and are starting to be used to try to understand about how gene variation leads to phenotypic variation in skeletal disease models. I'd like to talk to you a little bit about a scoliosis model that we've been working on. And this is an allele called DMH4. It leads to basically really drastic scoliosis where actually the bones of the spine are actually look pretty normal at micro-CT, but you get drastic dorsal and lateral curvature occurring. The interesting part, and going to our post-embryonic screen, is that actually the mutants do not affect early patterning of spine. So this is a calcine staining of 13-day-old fish, a wild type and heterozygous mutant. And you can see that basically the patterning is basically okay. It happens after juvenile development or during juvenile development into adult that you get this dysmorphology. When we clone this out, it turns out to be a missense mutation in the scospondin gene. And this was a very unique gene. The fact that when we got it, I didn't believe it for a while, and we sort of put it on the back burner. And it came to actually several different groups cloning the same gene for zebrafish scoliosis mutants, both in the lab of Ryan Early specifically. And what we found is that actually loss of function mutants leads to this sort of U-shaped larval phenotype. But we were not picking that up. We were picking out actually a pretty straight phenotype. And what we end up seeing is that in late juvenile development, we end up getting scoliosis, as we see down here in figure J. Well, we can make actually a loss of function mutation in this protein. This was work done in collaboration with Brian Serena in Toronto. And when we do this, we show that it really is a gain of function mutation or a dominant negative, and the fact that actually you can, when you lose the function of that particular allele, it reverts back to wild type, and that you see in K. So we have this mutation in scospondin, but it was unsure what scospondin was really doing because scospondin is actually expressed in this thing called the Reissner fiber. Now, those of you who don't know what the Reissner fiber is, don't feel bad because I didn't either. Reissner fiber is this extracellular sort of matrix that goes along the neurotube that involves with somehow sensory cilia interaction with the floor plate. And somehow this leads to scoliosis. And wonderful work by Claire Wyatt's lab has gotten into studying the urotensin receptors in the notochord or in the neural tube, and showing that actually, it's a sensory stimulus allowing for a lack of developing a straight spine that leads to the formation of scoliosis. And what we can see here in G and H is an antibody against the Reissner fiber. In wild type, you can see a wonderful, nice straight line that goes down the body axis and between in the ventricles down the axis. Whereas in the corresponding DMH4 mutant that we found, it actually started to get made, but doesn't actually continue on. So there's a defect in the extracellular matrix involving the formation of the fiber. Well, what you can do with this is actually quite interesting. So Brian's work has led to the idea that there actually inflammation that was involved in the mutant, and that this might be leading to the formation of the phenotype. And if you treat the corresponding mutants with basically this antioxidant and acyl L-cysteine, you can abrogate the early effects of the loss of function it's seen in homozygotes. So you see that above in F and compared to H. Well, that's pretty cool. The other thing you can do is you can actually treat mutants that grow up for a little bit and treat them late, and you can actually alleviate the late onset scoliosis that you see in these mutants. And this is in the heterozygous condition. You can see compared to the untreated versus treated, there is vast differences in the patterning of the spine. So all this work has been published in Current Biology, including the paired publication with Ryan Early's group, and Claire Wyatt's has published there recently as well. I recommend go look at these papers. They're quite interesting in terms of the number and frequency of the work being done. The second story I want to say that we sort of came across was a problem that exists and persists in the field, which is that there isn't a direct homology between teleost spines and tetrapod spines. And that can be shown by this, which is sensitivity to particular disease-causing alleles. In humans, one of the major genes involved in congenital scoliosis is TBX6. And you can see here a picture from New England Journal of Medicine, the sort of scoliosis that occurs in patients and kids that have these mutations. Well, when you make a TBX6 mutant in a zebrafish, the spine is patterned just fine. What you actually end up having is some of the arches get mispatterned, both in the ventral side, the hemal as well as neural arches get mispatterned, but the spine overall is fine. So I would say there's some disconnect between functionality, and this comes from basic biology of the formation of the spine between tetrapods as well as teleosts. So the tetrapod case, we all sort of, in textbook analysis, we know that actually cells migrate down and cover up around the notochord and start actually segmenting out in the pattern of presemitic mesoderm, the flow of segmentation that occurs. In teleosts, it happens much differently, which is actually the notochord actually starts to pattern first. And the sheath controls this. So the notochord, extracellular matrix around the notochord controls the early patterning of the spine. At that point, cells of the presemitic mesoderm migrate down and start forming the centra that follow the pattern of the notochord. So it's notochordal driven. That explains why TBX6, which is involved in presemitic mesoderm formation, is actually causing a phenotype in tetrapods, but not in teleosts. Well, we found a mutant that we called spondylus, and it formed a very unique phenotype, which can be seen down in the bottom right, which has these little hemisected centra. And so we're wondering about that and what this actually could actually apply to this question. Well, if you look back in evolutionary history, this is a progression of fossils of early extinct teleosts, or crown group teleosts, so they weren't teleosts yet. So all teleosts have what is this mondospondylus formation of their centra. Well, the lineage leading up to teleosts, actually you see a progression going from the fact that there's cases that have no centra, which is aspondylus, to diplospondylus, so they have actually two hemisected as well as two paired in the lateral row, to mondospondylus. What this mutation looked like it was forming was like an atavistic structure seen only in extinct teleosts. So when we cloned this out, we found that actually it's in this weird teleost gene called calminimin. What is calminimin besides actually a gene without many vowels in it? That's hard to pronounce. What we found is actually it's a dominant mutation that causes a gain of function effect. And when we make loss of function mutations in this, we can revert it back to wild type. It is due to a differentiation of an elastin-like gene in teleos after the whole genome duplication in the group. So basically, you go from this region that has an elastin, you duplicate it, and one of the copies gets a novel sort of thing put at the end, and this is calminimin gene. This is what is mutated. We don't see copies of this differentiated elastin within tetrapods. Interestingly, this gene is only expressed in the nodal cord, so suggesting that that actually may be the source of the instruction. The cool thing is when we assess this question of the activity of fused somites, one might argue is if we're reverting back to an atavistic structure, if we now test the function of fused somites, we might be able to replicate the congenital scoliosis that we see in patients. And we see just that. So above you see these are alizarin red fluorescent images. See wild type, see a nice patterning of the adult spine. Fused somites, it's patterned normally. You see a little change in your hemal and arches. Spondylus, you have this hemicentra formed. When you cross the two together, now you have basically an increased amount of fusions between the centra, and you can quantitate that, and it's very clear. So we think that what Spondylus is showing is that we now have a model in which we can actually test the early patterning events within zebrafish to assess how they relate to patients. And so we're following that up. So this work was also published in Current Biology just recently. So that's the for genetics component, but one thing we're doing is we're looking at one fish, and we're asking this one fish what it's doing. And so we're a little bit limited in that function. Well, we can also go and ask what happens in nature. And to do this, actually, we've developed a thing we call phylomapping to actually be able to track phenotypes based on genotype. Basically, the same thing you do in a mutant screen where we're taking a phenotype and we track the genotype. You go to nature and you say, these all have this phenotype. Do they share a particular genotype that follows suit? And so that shouldn't be very surprising. It goes back to Darwin because it's all about phylogenetic relationships. You know, I have traits like my father who had traits like his father before. Same thing with the fish. Can we trace it back in evolution? Problem is, even though there's a lot of sequencing in genomes, there's still a lot of deficiencies because you really need every step of the way to actually ask the question and trace the particular mutations that lead that phenotype. This is old data, about 5 years old now. But it's still a incredible deficiency that in fish now you probably have about 1,000 different species sequenced out of 31,000. And the same birds, we actually have a nice copy of a lot of different bird genus or genre, but the problem is that we don't have the species level specification within there, and so on and so on. So we need a new way to go about this. And so one way to do this is we came up with an algorithmic pipeline in which to use targeted sequencing. So we target pretty much all the coding, all the non-coding, and all microRNA within predicted within a genome, and pull it out of not only one species, but all their sister species that we can find that may have a trait, and then the ones that are ancestral that don't have it. So that is the reference 1 and 2 in the top right. And the way we do this, we make little algos against all these places, and we pull down fragmented genetic data, and we sequence them and then can realign them to make what we would call the conservome. So it's not the whole genome, but it's all the bits which we actually would use even if we had the whole genome to make an assessment. The key about this is we do it in pools. We take at least 5 to 7 individuals that are not related, we pull them together so that we can call basically variant SNPs that we see, as well as fixed SNPs within the genome. Because anything that is fixed should be leading to the formation of that particular trait that defines that species. So I'll give you an example that we use. We do this by making what we call the phylochip. I just explained a little bit about this, but what we can do is because there is relationships amongst a lot of these species we talk about, that we can use a pan, like even clade array, and pull out species from even distantly related fish because of this sequence conservation that they might have. So for example, species A in this phylogeny, we might use 50% of their sequences, but if B had sequences that were 15% different from A, we would add those on, and so forth and so on. So we get this sort of, we call seining, a large capture of all the genetic variation we can from a clade. And so this works amazingly well. So here's just a lineup between a species that's 70 million years distant from the RAG2 gene, which is the bar below, and one that's 5 million years. And the different little lines you see here are variant SNPs and whether they're captured in particular reads, which are the gray bars. You can see that 70 million years, there's a lot of variation seen in this particular species, but we still can align it back to the particular gene. That's the appropriate gene to look at. So we have this robustness about alignment because of conservation that we can do this up to about 70 million years. The key about this is that when you do this, you get a huge number of fixed SNPs because every species is their own organism, right? So I'll have a certain type of characteristics, but if I go over to my neighbor, he'll have other SNPs that define him as well. The key is actually looking between species and going down and be able to segment out what all these particular SNPs might have functionality. So you can start with all the fixed SNPs, go to ones which actually might be causing a change in protein structure, one in which we maybe can predict they actually might affect that protein, and then look for things like evidence of selection or clustering types of signaling or so forth. But the real power is if you do this multiple times. So if there's multiple species within a clade that share a character like big wings or long beaks or whatever you want to look at, then you can ask what is conserved between the species, between these lineages, because that has to be consistent for the particular phenotype. So the example I want to tell you today is about looking at fish that grow in the Antarctic ice. These are notothenioids. Here's a picture of one sitting amongst formation of ice in the Antarctic water. This is a wonderful study system because they show an immense number of different traits that are derived due to living at the lower temperature water that actually can actually be attributed to similar things happening in patients. And I'll tell you today about bone density, but there's also lipid accumulation in muscle. It's like David talked about. There's loss of heme in blood. And so we've touched on a couple of these, but the work we've done has really expanded the ability to use this model system to address functional variation in clinical analysis. Now, the story of the notothenioids is that about 50 to 60 million years ago, Antarctica started to drift away from South America, the Antarctic plate. And then there was a circling of current around Antarctica, which allowed for localization and a sort of insulation of temperature drop of the water in the sea. So you have high latitude and low latitude fishes that went across this. And what happened is that you ended up having a mass extinction within the Antarctic waters of species that were unable to accommodate this temperature change. And then a radiation of those that could. And of the ones that radiated, the notothenioid lineages are about 90% of the total biomass of fishes within the region. And so the phylogeny I'm showing you here shows basically where the radiation has occurred. And so what we asked is, could we use this phylogenetic method to actually get at the changes that we might see, genetic changes we might see underlying these trait analysis that go on. And this is work with Jacob Dane and Bill Dietrich, and they're at Northeastern. Also involved collaboration with Tom Neer at Yale. And so what we did is we got about 48 species and had pools of them, which we sequenced. And they exist from the phylogeny here. These are all the species we looked at. And this actually represents, about, what was it, about 10 to 15 million years of evolution. Now, when we use our array, let me get back. Oh, here we are. We got some of these fish and brought them to Boston Children and did CT scanning on them. What you can see is one of the fish of the outgroup here, Bovictus. These are high latitude notothenioids that they show sort of what would be sort of a standard bone density sort of spectrum of a fish. Well, if you look at one of these ice fish, at the same settings, you can see it basically they are massively devoid of mineralized material. And if you look at this, this is basically due to an osteopenia effect of very thin bones. And so if we look at the trajectory of bone density within the lineage, you can see here, and we put this out in sort of a phylogenetic series, that actually the basal groups have retained what would be a high level of bone formation. This is a heat map of basically of density we see here. But very quickly it's lost. And even at some of the more basal groups, do we see basically a very quick drop in detection of skeletal density. So we go about this, we made a file chip for the group and we added on ultra-conserved elements, microRNA, conserved non-coding to this. And we basically used primarily what was then the sequence genome that we had in place with this cordyceps, but we added little sticklebacks and a little bit of other species to that to broaden our analysis. And what we find is we can pull out about 90 to 95% of what we put into the equation. So this is a really efficient means of pulling out genetic material from this group. And you get a depth of about, anywhere from 30 to 50% semifinal. So we have enough that we can actually ask about variation. I hope I can change. There we go. And this shows analysis of CNEs. So these are conserved non-coding elements by genes. And you can see in a phylogenetically stratified sort of bar chart, you can see that we get coverage of all these. However, we see a distinct drop in sort of percent ID from about 96 to about 94. So about 2 sites per 100, only within the ice fish themselves. You can see that here. So the Elagenops is the sort of outgroup. Cryonodothenioi is the ice fishes, and you can see here that there's a variation of those, of the CNE average percent divergence, but it's clustered within this group. Outgroups themselves here are don't seem to have this variation. So it seems to be a distinct increase in variation in CNEs. And we actually see this pattern variation, and when we look at skeletal genes, we can go and ask which ones might we see sort of acceleration and drift in their sequences, which one have predicted function altering SNPs, and which ones might just be deleted overall. And this might give us an idea of pathways that are altered within these fish to ask about mechanism. And so we focused in on a couple of these, and in part because of existing models that we had in-house. One was on TRP11. TRP11 is a interesting cellular gene involved in processing of products in the Golgi. And you can see here a distinct drop within the nodothenioids and percent ID down to about 78% of the coding sequence variation. Distinct from even deeper variation you see even between stickleback and the nodothenioids. So something distinctly was happening within this signaling pathway. They no longer needed to maintain TRP11. There's no longer purified in selection on it. When we looked at TRP11, you can see here a progressive accumulation of SNPs that are predicted to be protein function altering. This is a Provian score here. What we see is that they're progressive and that you can see accumulation of these SNPs over time. So it fits with the hypothesis that this particular gene is going to drift. And this tracks very nicely with what we see in the changes in skeletal density. And when we make a model of this in zebrafish, we make a TRP11 knockout, that actually we see that you get in a incomplete penetrant manner, you get massive decreases in skeletal density within the mutants zebrafish. Now, the mutant zebrafish and nodothenioids are separate from about 250 million years. That's the last common ancestor. But since all these pathways are generally conserved, you can use these different models to test protein function. But what we see is that if this goes to drift, it's likely to also be involved with the loss of skeletal density. And so we know that TRP11 actually leads to lack of skeletal differentiation both in mouse. So a missense TRP11 mutant has basically a severe loss of skeletal density, but also that human fetus with TRP11 mutations basically have achrogenesis type 1A phenotype. And this work by Matt Warman and Patrick Smith in the Department of Orthopedics here at Boston Children's. But we also can look for epistasis. So then we look at some of these collagen genes we talked about, that you again see a progressive acquisition of predicted protein altering mutations within, for example, collagen 1A, 1A, right? And this also would be predicted to lead to somewhat of a osteogenesis imperfecta like phenotype, as we talked about earlier. When we model this in the zebrafish using some of our mutants, you can see exactly that. So two different mutations here leading to a decrease in skeletal intensity or differentiation within the adults. So the power is that we can actually start to pull out multiple different loci affecting the trait, similar as you would do sort of in a GWAS, but on an evolutionary scale. But But underlying this is a more fundamentally core statement, which is that the manifestations of disease that we think about in humans, well, they're actually regulated by the same laws which govern physical processes in general, right? So that, many conditions regarded as pathological in one species could actually be natural and selected for in another. And so I think that's what we're pulling out in many of these different types of scenarios, is that we actually get natural models of the disease pathology that we see in humans. That includes the spine, that includes bone density. And this is not a new idea. So I quoted this from a book from 1890. We are new to come back to it again. So I want to finish just by talking a little bit of this, just to bring it down to a close, which is we've done this, as I said, in notothenoids, which are right here. We've extended this analysis to over 250 species of teleos. You can see some of them here, both rockfish, snailfishes, sculpins, salmon, balaniforms, flying fish. And across the board, we see that we can actually pull down about 80 to 90% of the coding and non-coding sequence we target, and allows us to look at variation of these different novel phenotypes. And that is the plot on the right. And you can see here evolutionary distance to the outgroup. As I mentioned, for example, in notothenoids in one case, we've done balaniforms with about 50 million years, but we've also done rockfishes, which is even broader than that. So even out to about 100 million years, we're still able to pull out about 85% of what we put in by this method. And so we're extending these analyses to start looking at different case studies. And this is a life in extreme environments to actually ask if there's parallelism or convergence happening in different lineages to get the same type of environmental stimulus in the trait loss. For example, let's say bone reduction. Lake Baikal sculpins have almost identical phenotype as what happens to notothenoids in Antarctica. But what the question is the similar genes or similar pathways affected in these cases, which would argue that there's some common mechanism to actually allow for shift in phenotype. We're asking the same thing about snailfishes from the deep sea, other perciforms we get from deep sea, for example, eel pouts and Arctic fishes. We're looking at some Arctic char to ask these questions. And this is all work spearheaded by Jake Dane. But it's leading us to be able to do both approaches, which is to use a forward genetic approach to really ask about one gene, one function, to that of forward genomics, which is asked about pathway and diversity, about potential, and combine the two together to really get at a functionality and make, I would say, important, powerful models. So with that, I want to close. This is the lab. I put a little hash mark here around Katrin because she's now started her own group in orthopedics in Emory. It has been basically a combined and creative effort by everyone involved in the group, as well as a whole slew of collaborators who work with the naturals and work with us to make sure we're asking the right questions about the right type of organisms. And I'll thank my funding, Guggenheim Fellowship, as well as NIH and NSF funding for allowing us to do this work. And with that, I'll take questions.